in terms of the molecular, when do you get aggregation and when's that occurring?
Yeah.
No one really knows.
Okay.
And
So
that's
there's
the
not
same.
something that a lifestyle thing or a diet thing that creates this cell change.
There may well be, but we don't know what it is.
Okay.
There's no doubt, if, depending on the length of your glutamine tract, if it's above that threshold, you will get these changes. I suppose it's just about how aggressive, how early that occurs. And there may be lifestyle changes that you can do that would prevent that. But and that's the same for Huntington's, Alzheimer's, mad cow disease. Don't need be. There are, but no one really knows what those changes are. We can do many things in the laboratory. We can make it. We could probably add a whole range of metals, and they will speed it up. But they're probably not relevant to the things you're cooking your food with. So the answer really is no, we don't know.
How can you define what levels the ataxia goes? Like, for instance, I'm a paraplegic, but the next person might be a quadriplegic. So how can you define?
Okay, so with what we do, we can't. I think this comes back to what was said over here. That there probably are either environmental or there are other changes at the cellular level that are occurring which dictate the level of ataxia. And we don't know what actually, we have no idea what those are. All right? So you've got mutations in your, let's say, ataxin 3, but there may well be another protein or another cellular system which has also got mutations. And that means you may get the disease earlier. Actually, the converse could be true. You may actually get it later. You may get it at a different. But we have no idea. What we're beginning to understand for a whole range of diseases is there's the main protein, the main culprit. There's a whole range of other things that are going on. And what I do and we do in the lab is that's too complicated. We just like to work on one thing. We have enough trouble doing that. So we use a sort of reductionist approach. But there are other labs where they will try and look at the whole picture. In essence, they have to look in cells. But still, for almost all of the misfolding diseases I can think of, they believe there are other factors, other contributing things that are occurring, but we don't really know what they are. It's guesswork at the moment.
Can I ask, with your research, what's the next step in your research, seeing that you've now found a way of either increasing it, fastening it up, or slowing it down? Are you working with drug companies to come up with drugs to do that?
So we're working with the Victorian College of Pharmacy, who are experts in sort of the drug side of things, to take that forward. Where that goes, who knows?
And we don't know the time span.
The average time span would be, if you're asking when do we think we're going to have a drug, that's it. It's always 15 to 20 years. And that's if the things are working. What I think our work shows is up until actually worldwide, everyone was just focusing on the cues. And it's believed if you can get things to bind to the cues, you can stop these changes. The problem is as our work showed, there's 100 other proteins. And you can't target the specific one you want to. So with this mechanism we've generated, what it's done is at least open up another possible avenue of approach. And as I said, Huntington's, SCAR1, they've been shown to have similar mechanisms. So what this work's done is give us a chance of maybe getting something that will bind. But we're working at the protein level. There are researchers around the world working at the DNA level, trying to stop that sequence from becoming wrong in the first place. And there are others who are working actually at the final level of just trying to degrade obstacles and various things. So there's a whole range of approaches occurring. But it's always going to be long term. But I come back to something I said at the start. There's a whole range of these diseases. Mad cow disease, Alzheimer's, they all occur. They don't all have glutamines, but they all occur for similar reasons. Proteins fold incorrectly. And so, we haven't got treatments for any of those. But there's a strong chance if we get a treatment for one of them, we'll be able to translate that into the others. It might take a bit of time, but if it works for one, the rationale for the others will probably hold true.
You have explained this the situation as it is now, but somewhere in history, some field must have been where there was none of this occurring. How came that this all of a sudden Or how's it going and we have only just recognized it or
It's a
something?
disease. I mean, a lot of... Okay, so the spinocerebellar ataxias are not that common. They often, as I said, I'm not a clinician, but from talking to clinicians, we know they often get misdiagnosed for quite a while.
Yeah.
It was only in the mid-1990s did we understand what was happening at the DNA level, and that has allowed for diagnostic tests and various things to occur. So the disease would have been around for a lot longer than that. But what we do know is, you know, everyone here is more aware of the moment, is as you pass on from generation to generation, that glutamine tract can get longer. It actually can sometimes get shorter, but generally it gets longer. And so maybe it's a question of long, sort of, I don't know what I'm getting at, longevity or whatever, I don't know. I think it's always been around, it's just the numbers are so small, misdiagnosis a lot, is why it hasn't, I suppose, it's as prevalent as we think.
Yeah.
It's an incredibly hard thing at the DNA level and the protein level to work out what's happening and to distinguish one of these diseases from another.
Yeah.